Argentina. I have actually quite over the years had lots of connections with Argentina. First of all, I was trained in Tom Jovin's lab, who has obviously roots in Argentina. And a lot of the material I'm going to present actually originates from the work that I was doing in his lab at that time. And also I have people from Argentina working in my lab at the moment. And one of the group leaders, Hernan Greco, is also somebody that actually did a lot of development on the imaging methods to actually allow us to look at molecular reactions or biochemistry in living systems. So it's great to be here and present these results. So the Vice President also said that I should really keep to time, and I'm afraid he's going to throw things at me if I go over time, and I'm notorious for going over time, so please warn me, because I cannot see him behind me if he's going to throw some things. I can duck in time. Okay, so my talk will have the general thread, a red thread, which has to do with the fact that all living matter consumes energy to maintain organization. Everything is in constant flux, and that's a typical characteristic of life. And hopefully you're going to see during my presentation that you can actually exploit this knowledge to affect response properties and social behavior of cells by interfering with these systems. So I'm going to talk about a major oncogene. It is one of the first oncogenes found, called RAS, or rat sarcoma. And I don't think it's strange that it was one of the first oncogenes that was actually found, or oncogene product that was found. Because RAS, as you can see from this scheme, has a very central position in signal transduction. It actually integrates a lot of growth factor receptor signals from many different types of receptors. And then from there you get survival signals as well as proliferative signals that emanate from this node. Now, in order to elaborate on my talk and how we affect this system, maybe I should say one more thing. RAS is a major oncogene and is actually mutated in 30% of all human tumors, and actually in pancreatic tumors, 90% of all tumors have actually mutated KRAS. So there is, of course, a big deal to find inhibitors of oncogenic RAS. Now, pharmaceutical industry has obviously tried to work very hard to find these type of inhibitors, but to no effect till this day. And so this node, or this protein, has actually been declared undruggable. And hopefully I will show you that we might find other routes of actually affecting oncogenic RAS signaling. Okay, up to what RAS is about. So let's look first at the molecular level. RAS is basically a GTPase that runs in a catalytic cycle where when it binds to GDP, it's in the off state. You see that at the top of the figure on the left. And it needs an exchange factor, an auxiliary activity, to actually pry the molecule open such that the excess of GTP can then exchange and bind to the molecule. Now the switch regions will then organize in such a way that the molecule can interact with effectors and thereby propagate the signal. Now its intrinsic hydrolytic activity is very low. So it actually uses catalytic residues from another activity, another auxiliary activity, shown on the right side of this cycle, which is called a GAP, or GTPase activating protein, to actually bring it back to the GDP state. Now, because of this reason that it knows these two states, it has been called a molecular switch. But this implies that somehow that has a function at a higher scale. Now, this tells a lot about how the molecule itself functions, but very little about what happens in cells. So we have to look, in a way, at a different scale. So let's zoom out a little bit. We see yet less detail on the molecule. You see it now in the context of, let's say, the signaling pathways in which it operates. Now, what is RAS actually doing? RAS is nothing else than a switchable recruitment factor that causes proteins to translocate to the plasma membrane. Now, if you have a molecule like RA, for example, it's one of the molecules that gets activated when RAS is activated. Normally in the cytoplasm, its concentration in 3D is very low. And so that means that the activating activities like PKC that phosphorylate and activate it in the cytoplasm cannot activate it. However, phosphatases that have a very low Km value can actually very efficiently dephosphorylate it. That means that the cytoplasm, in a way, is an inactivating medium. Now, if you switch RAS on, you translocate the molecule, and because of the dimensionality reduction, you increase its concentration. And now the concentration comes in a range of the Km of the kinases. And thereby this reaction becomes very efficient. However, the phosphatases do not translocate. So that reaction remains as efficient as it was before, which means the battle is won in the direction of activity. Now, if the effector, the molecule that binds to the activated RAF, is itself an activator, like a GAF, then you get very interesting properties. So you get a positive feedback loop in the system, and the system can, on a population level, now truly exhibit a switch-like behavior. That means if you increase the dose of growth factor, at a certain point it will switch. And where this switch happens depends exactly on the amount of RAS that is at the plasma membrane. So controlling the dose of RAS at the plasma membrane is essential to control the way the cell behaves to growth factors. And that's what we're going to exploit. So the major question is, how is the spatial organization of RAS maintained? And how can we modulate it to affect its response, and specifically on oncogenic RAS? Now, let's look at the features that are relevant to the spatial organization of RAS molecules. And I show here two types of RAS, two major types. So the top one, Harvey RAS, HRAS, is both prenulated in blue, you see this little tail, as well as palmitoylated. So these are post-translational modifications that happens on the C-terminus in a hypervariable region that give this molecule hydrophobic properties. Now, with KRAS, it just gets prenulated. You just see this one blue tail, and on top of that, it has this polybasic stretch. I will talk in the second part of my presentation about this molecule because less is known about it. Now, if the cell would only have a plasma membrane, that would be the end of the story, because it's very simple. You make a molecule hydrophobic and it will just associate with the membrane that's there. Unfortunately, the cell looks like this. If you look at membranes, it's chock full of it. So the big hole you're seeing in the middle is the nucleus. The rest is all endomembranes. And so how is it possible that this molecule so precisely localizes in cells? Now you see it for HRAS very clearly at the plasma membrane and the Golgi, which I'll come back to in a second, and KRAS very clearly at the plasma membrane. So let's start with the palmitoylated RAS isoform. So let's say HRAS. Now the classical point of view of protein localization inside cells is about specific receptors. I think it comes from the finding that there are specific signal peptides that bring molecules to specific places in the cell. So initially, when RAS is de novo synthesized, it gets prenulated in the cytoplasm, and then the idea is that there's a receptor on the ER, the endoplasmic reticulum, that will bind it there. That's the first step. Then a second step takes place where another activity, a PAT activity, a palmitoyl transferase activity, will add these red lipid groups. Sorry if I can point at things here. There you go. These groups over here, and then it travels to the plasma membrane. Again, a receptor will maintain it there. And we spent several years to actually invalidate this simple static model and actually show that all is dynamic. So in 2005, we published a paper that showed actually that because this palmitoylation is reversible, it can actually be removed because it has this thioester bond instead of this thioether bond. It's constantly cycling around between the Golgi and the plasma membrane. However, at that time, we didn't have a clue why that was. We just knew it was happening. It took us then a few years to actually find out that this system is there to actually counter the entropic tendency of the system to redistribute RAS on all membranes. There is no receptor in the system. It's a dynamic system that maintains organization. Now, I'm going to show you a little movie that tries to capture the main features of the system. Okay, it's simplified, but it's kind of a sequence of events that simplified it. So the novo synthesized RAS has just this prenyl group. It moves fairly fast, and then it gets palmitoylated specifically at the Golgi. That generates a traffic jam. Okay, so you get a non-equilibrium concentration. The secretory pathway, these vesicles that you see, displace this traffic jam to the plasma membrane. It's still non-equilibrium. And now we get the major point, entropic leak. So the system will have the tendency to leak over all membranes, very slow. And the system corrects now for that, removes the palmitoyl group. The system moves very fast again. It can get retrapped at the Golgi, again a traffic jam, and then it's displaced by the secretory pathway. Okay, so the point is, and that's not intuitive, that even the palmitoylated molecule will eventually occupy all membranes. And the system constantly fights this entropic tendency by pumping back by the secretory pathway material to the plasma membrane. So in terms of rules, this looks like this. Palmitoylation at the Golgi is trapping, a non-equilibrium situation. The secretory pathway gives you directionality and displaces this traffic jam. That will eventually leak out because of the entropic tendency of the system, and so it needs a reset. It will depalmitylate the molecule. It moves very fast, such that it has a large exploration. Now, the last rule you're seeing is actually the most important one and the most generic one that allows us actually to affect RAS signaling by interfering with small molecules. Okay? The point is that this monolipidated or prenulated molecule moves way faster than this polylipidated molecule. Now, why is it that this depalmitylated molecule moves so fast? Okay, here you see actually a photoactivation experiment where we have basically made a mutant form of RAS that cannot be palmitylated. So it's like an intermediate in the cycle. And you see when we photoactivate it, this disperses very rapidly, but disperses aspecifically over all membranes. Now, if you think about membrane diffusion, it's very slow. It's in the order of something like, for peripheral membrane protein, micrometer square per second. However, the cytosolic diffusion is way faster, at least an order of magnitude faster. So the idea is there an accessory protein that binds this prenylated molecule and allows it to diffuse in the cytoplasm, such that it can explore the environment way faster to get retrapped? And so we looked in analogy for a molecule that looks like something that was known in the field of Rho GTPases and RAP GTPases, where there are molecules called GDIs, guanylucleotide dissociation inhibitors, that bind to the prenyl group, but specifically bind to the inactive state of the molecule because of this N-terminal helical region. Now, there was one molecule for which there was an overlay with this structure that looked very similar to a GDI, and this molecule called PDE delta. So the name is very awkwardly chosen, Phosphodiesterase Delta. It's not a phosphodiesterase. However, it was found as part of a complex of a cyclic JMP phosphodiesterase. That's why it's like a frozen accident, and the name remained. However, the structure looked very similar, so we thought, well, this could be maybe the solubilizing factor. So we had to actually verify if this molecule can bind to RAS inside cells. So does it really bind there? Now, for this, we used approaches where we can directly monitor interaction inside living cells. So what we did is we actually fused a cherry, a fluorescent protein, to PD delta, and we flew to citrin, another fluorescent protein, to RAS. And then we basically look at the FRET signal when in close proximity. So this is a very famous steep distance dependence of FRET that allow you to look at molecular interactions. However, the way we approach this is by using a quantitative approach called fluorescence lifetime imaging microscopy that allow you actually to determine the amount of interacting molecules inside cells. This was originally developed in Tom Joven's lab. So what you see over here on the left is basically the distribution of PD delta M cherry, which looks pretty homogeneous. Then we have M citrin NRAS, which is a donor molecule, and then you see a lifetime map. And there we have lower lifetime, you have interaction. This is semi-quantitative. But we can exploit the fact that each species, let's say the interacting species and the non-interacting species, have a discrete lifetime. So this is space invariant. The only thing that changes is the relative amounts. If you encode this a priori knowledge, we can actually make a map of the molar fraction of interacting molecules. And now this looks very interesting. What you see over here in blue means very little interaction. In red means high interaction. So this is molar fraction. You see it from the color table. Now we also know that in the region where you see the blue, so low interaction, is that it's there where we expect palmitylated protein, Golgi and plasma membrane. Whereas in the other area, we expect depalmitylated protein. So it looks like this PD delta is specifically interacting with a depalmitylated molecule. Now how can we prove this? For this we make use of an inhibitor that was developed in Herbert Waldman's laboratory in our institute that actually blocks the thioesterase activity, the depalmitylating activity. Okay, so this is the activity you see here on top. What happens then is that in steady state, the whole population is shifted to the palmitylated state. If now this PD delta cannot interact with this molecule, we should find a loss of interaction when we apply this inhibitor. So again, a sequence with lifetime imaging in live cells. On top you see a sequence of, or you see the distribution of NRAS, and initially you see at the lower, at alpha, basically map, you see that there's a lot of interaction in the cytoplasm. At steady state, there's a lot of depalmitylated protein. As time progresses, because we block the thioesterase activity, we see more and more palmitylated NRAS, and we see that we lose the interaction. So this really nails it. It shows that PD delta can only interact with a depalmitylated molecule. And this gives us basically the rules for the system that the GSF, we call it a GDI-like solubilizing factor, or PD delta, only interacts with the depalmitylated but farnesylated RAS molecules. And this is the most important part. It does this to enhance the kinetics of trapping at the Golgi. So again, this animation, now you see the leak here, the entropic tendency, the reset, thioesterase activity, and then PD delta moves it very, very fast, such that you can get retrapped at the Golgi and the cycle starts again. Now this sounds almost trivial, but it's absolutely essential. So if you downregulate PD delta in cells, you see what happens. On top, you can see the distribution of HRAS, where you see very nicely Golgi and plasma membrane, a beautiful distribution. You downregulate with scRNA PD delta, and you see this random distribution. Now, the exciting part was as follows. This also happens with KRAS. So KRAS, it doesn't have this palmitoyl group. If we downregulate PD delta, we see the same random distribution. So it's a universal factor to maintain organization. But we don't understand how it works with KRAS, so let me elaborate on that for a second. So KRAS has this prenyl group, this blue tail that you're seeing, okay? So that gives this hydrophobicity. But then you have this polybasic stretch. We don't have this palmitylation. Now normally, when it gets prenylated, it will just interact with all membranes in the cell. But the plasma membrane specifically has a very high dense of negative charge. So eventually, you expect there that you get an equilibrium situation where you have actually a concentration at the plasma membrane. The problem is it will take very long to equilibrate. So the idea was, wait a minute, we need here also this GSF to speed up the process such that it can be very rapidly trapped at the plasma membrane. So we looked again at the interaction between KRAS now and PD delta and got a disappointment. Because now if you look at the right picture where you see the molar fraction of interacting molecules, you don't see anything. There's no interaction. So now it looks like, okay, it's specific for N and HRAS, but not for KRAS. However, this was wrong thinking, because the molecule helps to maintain it. So we're not looking at the right population here. Maybe what it's doing is actually removing material from endomembranes. So what we need to do is actually remove KRAS from the plasma membrane, bring it back to the endomembranes, and then see if there's an interaction. So we apply another, let's say, chemical trick. And here, we put in an activator of PKC that's known to phosphorylate a serine that is close to the polybasic stretch. So now we add quite a bit of negative charge that partially neutralizes the positive charge on the molecule, so you get some repulsion. Yeah? This will redistribute material towards endomembranes. Okay, let's see what happens. Now, on the left, where it says pre, you have no inhibitor, nothing there, or no activator, sorry. You see a very nice plasma membrane distribution, and then we see no interaction whatsoever with PD delta. But as the situation progresses, you see more and more material now diffusing into endomembrane system, we see more and more interaction. Now, I've on purpose put the wrong description. Is it really an extraction, or is it a passive process? Does PD delta simply solubilize anything that's in the cytosol? So the question is really, do we have a process like this, where it extracts it? Or do we have a process like this, where we have an equilibrium with very little soluble prenylated material that then binds to PD delta and just shifts the equilibrium? Now, in order to answer that question, we have to do quantitative biology, where we determine kinetic constants in living cells. Now, in order to do that, we resorted to a mathematical approach called Cellar Automata that allow us to actually simulate reaction diffusion in a realistic cellular geometry. What it means is this moving picture that you're seeing, we take with the microscope basically a three-dimensional reconstruction of let's say, the relevant compartment where this molecule is. And then apply the rules that I've shown before using the Cellar Automata to do the reaction diffusion. So Cellar Automata is nothing else than a discretization of space, time, and state, where the state of each cell, each part of the raster, is determined by its environment in itself, the future state. So it's very easy to intuitively implement the rules that I've shown before. What's even more powerful, you can use this approach not only to test hypotheses if it is compatible, let's say your hypothesis of how the dynamic system regulates spatial organization, but you can also actually fit